Thanks a lot. Thanks a lot, Rita. And as I said before, I'm really sorry not to be in Bologna because that's a city that I love. I like a lot, and it's almost one of the highlights of the year, right? When we come, we teach, and we see the friends, right? We see and Rita, Emilio, and many others. I don't know who is here today or not, but it's and then the dinners and the talks and the conversations and the interaction with the students. So all these things that we have lost during last year, as I was saying before, my last international trip, and I have taken only one flight since a year ago, was to Bologna, which was at the time when everything was starting and we had no anticipation of how this thing was going to evolve. So I will talk about something. I don't know whether this is what Rita expected from me, but anyway, I will talk about something. Let me just share my screen. Some of the things that Rita had mentioned. Do you see my screen?
Yes.
Okay. So you see now better? Yeah.
Yes, Adil is in presentation mode.
Okay, good, great. So I will talk about. Yeah, I'm sorry, I didn't prepare anything about sex. I saw that this topic, sex, was sort of a little bit delicate, and maybe next time I will promise that I will talk about sex. I will talk about death today, so that the two main interests about probably in people's life. Anyway. And as I said, I mean, these things have changed. I just want to emphasize, my last trip was before the pandemics, and there has been a lot of research in, and this is something really amazing, right? There has been a lot of huge effort of all the scientific community that has been working on topics related to COVID-19 from many different angles, and the number of papers that have been published in bioRxiv and in journals has really skyrocketed. And I think it's a great demonstration of how willing we are to adapt our research interest to problems that actually had a real impact in human life. And this is actually the way in which this should be, right? We're trying to understand the world, to make the world a better place to be. And now we had an opportunity, most of us as biologists, to contribute, even if it's minimally, to a better world. Unfortunately, I'm lazy, maybe, and I haven't done anything related to COVID-19 or SARS-CoV-2. But I had just one slide that I like to show, which marginally touches on the issue of COVID-19 and research and democracy, right? So this is a plot that I made several years ago, in which the dots corresponds to the countries, and what you see at the bottom is a democracy index, whatever it means. Larger the country, more democratic, and in the y-axis we have some sort of scientific quality of the country, how good the country is in terms of science, and this is measured in a simple way, maybe silly way, which is just the number of publications per capita. And what you can see very clearly, it's that there is a correlation, right? There is a correlation. The countries that are more democratic, and also the countries in which there is higher, better science, right? Let's say that there is this correlation: more democracy, better science, and this is actually even this correlation even exists if you correct for research spending, because you may think, well, of course, the more democratic countries are the richer, they are expending more in science. It has nothing to do with democracy. It's just why is the amount of money that you spend in science. But if you correct for this using partial correlations, you still see this association. So at the same level of investment in science, science is better in a more democratic country. Of course, there are exceptions like Singapore, right, that they did a lot of, and they actually for a while were able to manage a high level of research investment in a country which is not particularly democratic. And then I say, you may ask, why are you bringing this? What does to do? What this thing has to do with COVID? Well, then there is this thing, right? This is a little bit old, but anyway, I mean, I don't know what it. Well, actually, I'm sorry, Italy and Spain are at the bottom of the rank, if I must say. Anyway, this is, I don't know how reliable this thing. I don't want to make this a strong point, but let's say that there is some truth, right? There were a number of countries in Europe that responded well to to the COVID-19 crisis, and there were other countries that responded worse, right? For whatever reasons. And then what I did, I just plot these countries into the previous scatter plot, right? And so these are the countries that that responded the better to the COVID crisis. And as you can see, I mean, these countries tend to be to the top, to the right. So these countries are countries in which there is more democracy and there is more research. And where are the countries that were responding the poor to the COVID-19 crisis? Well, the countries that responded the poor, with exceptions, of course, were actually on the other end. They were countries that were less democratic and countries that invest less in research. So what I just wanted to point here that there is a link, right? I mean, science is not something that science is not something that's isolated of the environment in which it is produced. Right? You may think that science is just in a question of investment and putting resources, but science is also science tends to flourish in the places in which open debate and discussions and the capacity to challenge the authority, which is not very common in the countries of the south of Europe, right? Where the professors, we are all in a pedestal, and if a student disagrees with us, we tend to dislike it, right? And this doesn't really happen in other countries and other cultures in where actually it is almost an obligation if you're a student to disagree with your boss, right? Because then it shows critical thinking. So there is this relationship between society, science.
Excuse me, Roderic.
Yeah.
Excuse me.
Yeah.
This is Rita, obviously, interrupting.
Yeah.
Sorry for
Yeah.
this.
No,
Can you give us a flavor of how the democracy index is computed? So to say, what is democracy here?
Yeah, no, this is a very good point.
Because in Italy, I'm always claiming that there is too much democracy at the moment.
Well, not in this index, actually. Italy and Spain are next to each other.
Mm-hmm. So what is democracy?
Democracy index, there are many democracy indices. This is, I think, is taken, I mean, I am very skeptical about this democracy index. I think that this is taking, I need to check, this is taken from The Economist. The Economist has an index which is based on, I don't know, it's about 20 or 25 different variables which have to do, let's say, for instance, free speech, how representative the institutions are in terms of how the elections, how many referendums are in the countries, right? Maybe for that reason why Switzerland is here. I don't know. There are many parameters.
As to free speech, we should be the first in the world. Okay.
Well, this is weighted, but I can send you the link of how this is on and which parameters. And I mean, this is very, very, very approximative, right? And I mean, it's just an idea, right? It's just, I think what I wanted to mean is that the better the society, the better the science in this society, the better prepared the society is to react against threats. And that's what I wanted to point here. And actually, because I wanted to say something about COVID-19, I know that that's a little bit far-fetched, right? But I think it's still something that could be that could be said. So let me move to my talk. So actually, this week, by the way, and as yesterday, right? This is February 16th. 20 years ago, the draft genomes of the draft sequences of the human genome were published. Right? They were published. There were two drafts published independently, one by the public consortium in Nature, and one mostly by a company called Celera Genomics that was created during the Human Genome Project to accelerate as an opposition to the public project, because Celera Genomics was funded by a scientist called Craig Venter, who actually claimed that the way in which the public consortium for sequencing the human genome was too slow and too expensive, and that there were other ways in which this could be accelerated. And he actually created the company, Genomics, to sequence the human genome. And the first drafts of the human genome were published in these issues of Nature and Science, as I say, exactly 20 years ago this week. Science 20 years ago yesterday. And this, by the way, if you look at these issues, I mean, these are the largest by far papers ever published into these two journals, right? They are almost 40 pages long or more, right? Which is unheard of in Nature and Science. And I just wanted to say, I did a very, I just up to this because I had, to some extent, the privilege, sort of luckily, of participating into this project, and mostly with the Celera Genomic Project, and in the middle of this, many names, the names of, I don't know where we are, but I think we are here. So the names of a graduate student of mine, Giuseppe Fabril, and myself are here as authors of the sequence of the human genome. We did really very little, right? What we did for this project was just producing, we generated, we had a program called GFF2PS that was able actually to display the annotation of arbitrarily large genome sequences. And so arbitrarily large that our program was able to actually display the annotation of the entire human genome, right? And that's actually what we produced, the poster that was distributed with Celera Genomics. And I just wanted to mention something, right? This is, as I look back, like 20 years ago, I recall these days that were very, very exciting, right? We were actually went to Celera for, I think, 2 or 3 weeks, I forgot. And this was really maybe the most exhausting weeks of my life, because we spent the entire day working on this, because it was very difficult to produce this poster. I mean, you don't really understand. We are plotting here 18,000 genes with exonic structure of the genes. And this is in A0. And it was almost impossible. So we had to develop a lot of software and dynamic programming methods, etc. And we wake up early in the morning and stay until very late. And we had meetings every morning with the entire team that was doing the analysis in the Celera team. But really, this was so satisfactory, right? This is something that I really want to emphasize. When you work on something that you think it's important, really work is a pleasure. And these days I'm reflecting on this, because I haven't done anything on COVID-19, but I'm thinking of the people that have been developing the vaccines, right? So much stress, so much work, so much enthusiasm, so much drive that I actually envy them. I would have liked to work on this, right? To be able to participate on something that is going to have such a big impact in the life of people. And the point that I wanted to make is essentially, at the end, it's the same effort if you work on something important that if you work on something less important. And the difference is that if you work on something important, you really enjoy yourself and you feel actually gratified and you really self-realize. And then I would recommend you to work always on things that are important. Now, of course, it's very difficult to know what is important or not. Right? People working on retroviruses before the HIV, they were working on some odd area of science. I think that the point is that you have to believe that what you're doing is important, right? So if do it the other way, when you work on something, really convince yourself that this is important, because you will really enjoy it much more. Anyway, the Human Genome Project was the culmination of a project that lasted for more than 10 years, probably, right? It started early in the late 80s, early 90s. It was mostly funded by the 2 institutions from the United States, the National Institute of Health, NIH, and also the Department of Energy from the United States. And it also has some important contribution from the Wellcome Trust Sanger Institute in the UK. These were the main contributors to the Human Genome Project. And well, there were many landmarks during this project that culminated, right, as I said, in 2001 with the with the sequencing of with the release of the first drafts of the human genome sequence. You may not remember because you are very young, but that this was announced, right? The announcement of that the humanity actually had been able to decipher the sequence of the human genome was announced simultaneously by the President of the United States, that was Bill Clinton at that time, and for the Prime Minister of the UK, right, that was Tony Blair, in a joint conference. And there were no Zooms at that time. We're talking about 20 years ago, but that's the close thing to a Zoom, right? The teleconference, right, in which they were both presenting simultaneously from Washington and from London, right, the news that the sequence of the human genome had been obtained. I don't recall really any other, maybe of course, maybe with the exception of the man reaching the moon, the humankind reaching the moon. I don't recall any other scientific discovery that has given so high political relevance as the sequencing of the human genome. And here you see Craig Venter, right? This is as I said, this is the founder of Genomics, which sequenced one of the drafts of the human genome. And this is Francis Collins, who was the director of the National Human Genome Research Institute, which was the institute that led the sequencing of the human genome from the United States. And now Francis Collins is the head of the National Institutes of Health in the United States, so one of the top advisors to President Biden in science. So there was a lot of hype, right? Why there was this relevance given to this discovery? Well, because it was the anticipation. Well, this is just a few years. This is a couple of years, or 1 or 2 years before, maybe a year before the release of the drafts of the human genome sequence, and of course the expectations were great, right? Were really extraordinary. So, for instance, in this article in the New York Times, it says healthcare will shift from a focus on detection and treatment, right, which is what we do still in disease, to a process of prediction and prevention. So the idea is that we will not get sick because we will be able to anticipate which diseases we are likely to suffer, and then we will take actions before the diseases show up, right? Rather than what we do now, or we were used to do, which is detect, and when we are sick, we will do treatment now. And you say, you can imagine having an infant tested at birth, and the result says that you are susceptible to disease A, B, and C. Okay, so this were the expectations, and I can to tell you that there were people, scientists, respected scientists, that in early 2000, they were claiming that in 10 or 15 years, people will live to 120, 150 years old without major complications, right? And this was maintained at that time. The truth is that things turned out to be much more complicated, right? And there's actually an article in you remember, right, this paper that say, imagine tests being tested at birth, right, blah, blah, blah. And now this is an article 10 years later, I think, yes, in 2009, just 10 years later, in also the New York Times, which says genes show limited value in predicting disease. So the era of personal genomic medicine may have to wait. So the point that I wanted to make is that there were a lot of expectations that were made around the initial sequencing of the human genome, and that these expectations were, at least after a while, not met as largely as it has been expected, right? And I don't want to. I mean, and I know, okay, and I know why this is. I think there is a missing slide here. I know why this. I know. It's not that I know why it is, but we can guess why this is, right? So the thing is that the genome, the rationale for sequencing the human genome was that all the biological traits of organisms are encoded into the sequence of the genome, right? That's what we expect. So a frog is a frog, and an eagle is an eagle, and a rose is a rose, because their genomes are different, right? If the genome of a rose was identical to the genome of a frog and a rose will be the same. right? So the biological traits of the organisms are encoded into the genome, and also the biological traits that make individuals different from each other. So the color of my eyes, of my skin, my height, I don't know, the color of my hair, the predisposition to suffer particular diseases, some psychological traits, and many things. Essentially, many biological traits, or most biological traits, are encoded into the sequence of the human, and the sequence of the genome, right? In the case of the human, the sequence of the human genome. And this is true. We were not wrong about that. This is what we still believe. I think most scientists believe that the large fraction of the biological traits of individuals are encoded into the sequence of the genome. The problem is that the way in which these traits are encoded into the sequence of the genome is mostly unknown, right? So it was not as easy as, well, we'll have the sequence and now we'll know, right, how the sequence encodes its instructions, because these instructions are encoded in a very complex way, right? And this will not be the topic of my talk today, but you can imagine, right? All the complications that, how complex is a process in which from a single cell with a genome, right, an individual is developed in 9 months, right? The huge number of cell types, the connections between the cell types, the organization of the cell types into tissues, into organs, into the systems, into the final body, right? How complex is the interpretation of these instructions, right? Anyway, obviously, after the human genome sequence was released, there was the need to know what to do with the genome sequence, right? What to do with the genome sequence. As I said, the realization was that we did know very little about how the information was encoded into the genome sequence. And I could say that there was a number of post-genome, I'm talking to, and I will just try to, as this is my view, things have gone after the human genome sequence, where the efforts has been put into this sort of like post-genome research. The first thing is the following, right? The draft sequences produced by the public consortium of Barcelona Genomic were just sequences of one individual. Well, actually not even one individual, were single sequence. In some cases was the mixture of the sequences of few individuals, we don't know well, right? But essentially it's one sequence, right? Or two sequences if you want, one per each project. But of course, we are different of each other because our genome is different between us, right? So this human genome, right, does not belong to anybody in particular, belongs to just one person. It is very difficult, right, to know how variations in the phenotype between individuals relates to variations between genomes of these individuals if we do not know, right, the genomes of more than one individual. And actually, I remember what is the slide that I missed here. I just wanted to say that the sequencing of the first human genome took about 10 years and cost about hundreds of scientists, and it cost $3 billion, right? $3,000 million. So it was impossible just right after the sequencing of the first individual to assume that we are going to sequence 2, 3, 10, whatever, right? But anyway, it was very clear that there was a need to characterize genome variation, right? The second area in which post-genome research has proceeded is into the understanding, right, how genome works. So a number of projects have been developed just to understand how the instructions in the genome, all these at molecular level, are encoded right into the genome. And the third great area is actually the fact that we are not the only organism on Earth. I mean, there are many millions of species living on the Earth. And there's also an interest for many reasons, right? From the just the egoistic point of human health, because many of these organisms are or could be disease models, but also, for instance, in terms of maintaining conservation of biodiversity, etc. So I will touch briefly, right, on these three avenues, focusing mostly on what has been the work of our group during these years, which is understanding genome function. So one thing, if you have questions, I cannot see the, why I cannot see the hands? Well, I don't see the hands raised, but anyway, if you have questions, just raise your hand or interrupt me, and I will be happy to answer them, whatever, so that we'll make this a little bit more lively. I don't know if you have any specific question. I mean, I have material. I think it's not important if we don't finish everything. It's just more important if we learn together, right? So if you have a question that you want me to answer now, I can stop or I can go ahead. It's up to you. The only thing is I don't see. Maybe you are controlling this, I assume, right, Luigi?
Yes, I can. I can control the.
Okay, perfect. If because I don't see any. I don't see the raise hand thing. Maybe because I am in full screen mode and whatever, and I am presenting. Okay, good. So, but you are Pierre. Don't hesitate to interrupt me if it is a question.
Sorry, it is under reactions. Okay.
Yes, but you cannot see because it is presenting for all the other people. Raise your hand under the reaction menu. You can find, or you can ask in chat. I will repeat to Roderic.
Great. Thank you. Okay, so first thing, so there was, as I said, and all this research, although I'm not really explaining this, but I think it's very important. All this research has been possible because of impressive technological advances, right? So there has been mostly from mid-2000s, right, the development of these next generation sequencing instruments. Now I don't know really, but now, I mean, as I said, remember that I said that the cost of the human genome was about $3,000 million per genome. Now I would say that the cost of sequencing a genome is maybe $500, right, or 500 euros, more or less than 1,000, and probably the cost would still further drop, right? So this is what has been. This is what is actually making this possible, as well as the development of not only of instruments, right, and of the protocols and of the strategies that using these instruments, not only to sequence the genome, but actually to measure the activity of the genome through the functional genomics assays like RNA-seq or ChIP-seq, whatever, in which you actually don't sequence directly the DNA, but actually sequence things that bind to the DNA, that actually or that originate from the DNA, that are actually the cause of the action or the consequence of the action, right? So you think that you should ChIP-seq, you are able to identify fragments in the DNA in which transcription factors bind. Through RNA-seq, which is actually which triggers transcription, through RNA-seq, you're able to sequence the RNAs being produced by the DNA sequence. So the output, the result of the action of the genome. So, I mean, through all this, right, the technological developments in the instruments of the protocols, we are really advanced a lot on our understanding of the function of the human genome, which doesn't mean that we are even closer to the full understanding. Anyway, so I just very briefly, so there were I think that one of the first projects after the Human Genome Project was called the HubMap first, and then which was based on genotyping, not sequencing, and then the 1000 Genome Project, in which the idea was to characterize human variation. Now, know that there is I think this is important, there is now a concern that many of the applications, in particular medical applications, that are derived from genomic studies are based mostly on genomic data from the Western world, right? From mostly individuals with high European ancestry. And it is very well known that the ethnic background may have an impact on the way in which the genome reacts to diseases and to cures, right? So there is this effort into trying to be as comprehensive as possible with projects called like the Genome Variation Project, the 1000 Genomes Project, in which actually the aim is to have really an exhaustive representation of the genome variation across as many human populations as possible. And this, I mean, and these projects, right? These projects, this these projects are at the seat of what had actually evolved into the personal genomics projects, which you probably have heard, right? These are actually not so much research-oriented projects, although really the differences between research and medical practice are very blurred in the case of genomics, because any genome is an object of research, right? Any genome can be used for research, right? And any genome from individual have some sort of medical value. So there are all these personal genomics projects all around the world, and then maybe the most advanced one is the 1,000 genomes in the UK, where now the genome of more than 1,000 individuals has been sequenced already in the UK, and the plan is to scale to a few million. But there are initiatives all over the world. There is also an initiative pan-European initiative to sequence the genomes of about 1 million European citizens from different European countries. So we do, and because of importance in medical, I can't say, I don't know how many human genomes are now sequenced, but probably it's the order of millions, right? Many of which actually not going to end up in research. Or maybe yes, but I mean, many of which are obtained primarily for medical purposes, not for research purposes, right? And this is, of course, an area that will grow in the future, because this will end up being occurring in every country. And of course, there is a lot of bioinformatics and medical informatics that's associated to this, because the important thing, of course, is to relate the genomic sequence with the phenotypic, the clinical, the medical information, mostly in electronic health records, and to link this as well with all the instruments now that we are using now, like these watches and these phones, right, that are tracking, right, all the time my physical activity, and also some physiological measures like, I know, the heart rate and things like that. So there's here a huge area of possibility of need for informatics and bioinformatics developments. So the second area in which, after the human genome research, after the human genome draft sequences were published, was that the projects that aim to characterize the function of the human genome, right? What is called the functional genomics projects. So one of the most important of these projects, maybe you have heard about this, is the Encode project, right? The Encode project is a project that actually started in 2003 with a pilot phase. So just for here, have you an idea? First, the pilot phase of the Encode project started in 2001. And because of the technologies that although we did know the entire human genome, published in 2001, that's because the technologies that we had available at that time to interrogate genome function were really limited in capacity. The Encode project limited to 1%, to the analysis of a 1% of the human genome. I think it were 33 different regions, more or less on the different chromosomes, that were selected for different criteria, and that actually were deeply analyzed in the first phase, the pilot phase of the Encode project. Now we are in the fourth and the last phase of the Encode project, which actually will end up this year, or maybe next year, or early next year. Anyway, it would have lasted almost 20 years. It is most likely the genome project, the functional genome project that has lasted the longest, even much longer than the human genome, much longer than the human genome project. So the goal of the Encode, of the Encyclopedia of DNA Elements, is to identify all elements in the human genome that confer functionality. Of course, the functionality of the human genome is mostly encoded into the genes and the transcripts, right? These are the genes, and the transcripts are the regions of the genome that are transcribed to RNA and then are eventually translated to proteins, and the proteins are the main effectors of biological function. So in the case of the human genome, less than 2% of the human genome actually ends up being translated to protein. There is now a wealth of other transcripts which were unknown when the Encode project started, it's mostly unknown, that are called long non-coding RNAs. These are RNAs that look like protein coding genes, but are not translated to proteins, and they rival in number now to protein coding genes. So there are about 18,000, between 18 and 20,000 protein coding genes into the human genome, and there is probably a similar or much larger number of long non-coding RNAs. So just, I mean, something that maybe may surprise you, right? So we are 20 years since the publication of the sequence of the human genome, and we don't exactly know how many protein coding genes, not even less than long non-coding, are encoded into the human genome. We have settled in a number which is between 18,000 and 19,000. I forgot now whether it's closer to 18,000, closer to 19,000. This number is quite stable, but if you look at the main database, the main repository, and the main resource for human genome annotation, which is GenCode, which there are releases every 3 or 6 months, you will see that there is a change in these numbers. Maybe it's 2, 3, 4, 10, 15 genes that change. So what is still unclear, how many protein coding genes 20 years after the completion of the human genome are encoded into the sequence, right? But genes and transcripts are not the only regions that confer functionality, because there are other regions of the genomes that are involved in the regulation of the transcription of the genes, right? The things that actually confer cellular specificity, the fact that cells are different one from another, is not that they have different genomes, right? The cells in my body, they all have the same genome, right? The cells of my skin have the same genome that the cells of my eye, than the cells of my bones, but the cells are very different. They are very different because the RNAs that are produced there are different. Some genes are expressed in one cell type, some genes are expressed in another cell type, some genes are expressed in two different cell types, but at very different levels. So there are many regions in the genome that, although don't directly code for genes and transcripts, are involved in the regulation of the expression of the genes and transcripts, and these are proximal elements like the promoter-like elements, and or distal like enhancer-like elements, and then all these are of course regulated by many factors. And essentially, the idea of the encyclopedia of DNA elements is trying to characterize, right, these elements. And there is a thing now called the CCRE registry, which is called the Conserve Candidate Regulatory Elements. Conserve candidate regulatory elements is here, which is about 1.2 million, 1.5 million, which is the outcome of the last phase of Encode 3. This is maybe you see it here in the cover like it's 3. Because this is the third phase of Encode. The papers, a number of papers were published actually just in summer last year, and among these papers, right, there is this paper that describes this atlas, right, this encyclopedia, because the aim essentially of the Encode project is to identify all regions that have functionality. We did actually participate in Encode 3, and I will show you briefly what is the sum of our contribution to Encode 3. This actually was we published it's called a companion paper. We published this in Genome Research. The cover is a cover that we propose to explain or to illustrate what was the meaning of our research. By the way, this is a paper, the first author of which, Alessandra Breschi, is actually a student of your program. Right, this is one of the students that came from the Bologna Master. She's a very, very good student. And she was obsessed to go to work in a company in the United States. And after actually finalizing the PhD in my group, she went for a postdoc to the group of Mike Snyder in Stanford. Mike Snyder is one of the top names in the field of medical functional genomics. And then I think she's now working in a company in the Bay Area, which was her dream. So I'm happy that she was able to fulfill her dream. Anyway, this discover what it means is that discover is based in a painting by Picasso. It's called Belle Before a Mirror. And there is a young woman who is actually looking herself into a mirror. Into the mirror, she sees as a sort of deformed or less appealing view of herself. And there are many interpretations of this painting. One is that she sees herself as an older person, right? Here, actually, we are interpreting this as she sees herself as a diseased person. She is sick, and then she sees the reflection of her body as a body of the sick person. And because essentially one of the things that we explore into this paper is how the cellular composition of the organs may reflect changes in the cellular composition of other organs may reflect pathological states. And I will develop this over the entire talk, right? Just what we did in Encode is relatively straightforward. Tom Gingeras from College Harvard. This is many years ago, right? I mean, I don't think I'm lying if this project started maybe 7 or 8 years ago. And it took forever to publish. That's the problem with large scale projects, that everything gets very, very, very slow. Anyway, what we did at the time is, Tom did actually, is he selected a number of primary cells from multiple body locations, right? So this data did not exist at the time. So he just took primary cells that have some sort of, let's say, just to be superficial, primary cells are cells that have some sort of common morphology and function, right? They are, let's say, groups of cells that are very similar. And he took actually primary cells from many different organs. I think it's about 60 different primary cells from, I don't know, maybe 10 different locations in the human body. And then he did RNA-seq, so he sequenced these populations of cells. So one thing that I want to emphasize, I think this is known for all of you, but I want to emphasize this because it's sort of important for what I wanted to mention later on. So most RNA-seq, until very recently, when you wanted to do RNA sequencing, the quantity of RNA that you needed to do the experiment was so large that you didn't have enough with one cell, right? You had to have millions, hundreds of millions, maybe billions of cells to extract enough RNA to have an RNA-seq experiment. Therefore, when you have an RNA-seq experiment, and then you measure the gene expression of genes in this RNA-seq experiment, you are not really measuring the actual number of copies of this gene in a particular cell, right? When you get, I don't know, RPKM of 10 or 15, this doesn't really correspond to any single cell. Actually, it may actually be that in your population of cells that you are sequencing, there is not a single cell that has 10 RPKMs. It may be that half of your cells have 0 RPKMs, and the other half have 20 RPKMs. But because you extract the RNA from all of them, at the end you measure 10 RPKMs, right? So what you get is an average of the expression of the genes across the millions of cells that you have explored. Of course, this is less important here because, as I said, these are primary cells, and we had this intuition that these cells are quite uniform. Anyway, what we did after doing this RNA-seq, this expression RNA-seq, we just quantify the genes, and then we just compute this sort of correlation of expression across the different experiments, different samples, based on expression. So essentially, I know if we have 20,000 genes and we have 2 samples, right? We compute the correlations between the expression of these sets of 2,000 genes, right? And we get correlation. Of course, here in the diagonal, I am correlating the expression of the genes in the sample with itself, and the correlation is always 1. Right? And these are the different samples. As I told you, it's 50 or 60, I forgot, between 50 and 60 samples from different body locations. And the colors here, right, the colors at the top correspond to the different body locations, the lung, the skin, right, whatever, the heart, whatever thing, right, the kidney is here. And but what you see is the following, right? I mean, maybe with a little bit of imagination, you actually see some structure, right? You see a very large cluster here, right? You will see a very large cluster here of samples that show some sort of similar transcriptome. You see here 2 samples that show a very similar transcriptome different to the rest. Here you see again another group of samples that had a very similar transcriptome. And then here you have another group of samples, or maybe 2, it depends on how we're going to see it. Let's say 1 group of samples that also have a very similar transcriptome. And you may think, well, maybe this is reflecting the body location, right? I know the cells from the heart, they are similar and clustered together. But actually, if you look into this, right? If you look into this, you immediately see that the clustering doesn't reflect any body location, right? There is nothing clustered by the body location. Actually, we have the germ layer here, although that's not very good. The germ layer here, and they don't cluster also from embryological origin. The way in which the cell, what are these clusters, right? All these clusters are the following. So all the cells here are endothelial. All the cells here are epithelial. These 2 cells are melanocytes, some reason clustered apart from the rest. And the cells here are sort of a high variety of cells that include fibroblasts, muscle cells, mesenchymal cells, adipose cells, skin cells, etc., etc., etc. So we had, well, not actually skin cells, anyway, breast cells, etc., etc., bone cells. So we had like, let's say, let's forget about melanocytes that are apart, right? We have essentially like 3 major group of cells. We call them epithelial, endothelial, and to this group we refer as mesenchymal globally. And actually, the thing is that when you try to see to this clustering, what is the contribution of the body location of the embryological origin, we actually saw that they contribute very little to this clustering. So primary cells don't seem to know in which organ they are, and don't seem to know what is their embryological origin. They cluster by some sort of function, right? By some sort of function. And we go into this and you ask, where are, for instance, the cells of the lung in this clustering? Well, the thing that you see is the cells of the lung are everywhere, right? The cells of the lung are here, are in all the groups, essentially in all the groups, in epithelial, in endothelial, and mesenchymal. So maybe to an extreme, what this is telling us is that within an organ, there is similar transcriptional complexity than the entire human body, right? That's essentially one way of seeing this. That I can find 2 cells from the lung, right, that are more dissimilar to each other than a cell from the lung and a cell from the kidney, right? I could find 2 cells, one from the lung and one from the kidney, that are more similar to each other, albeit from the transcriptional standpoint, than 2 cells from the same organ, from the lung, or 2 cells from the liver. Okay? And actually this is recapitulated when we use other data sets. This is data from the Phantom project. This is actually another of these. This is another of these functional genomics projects that I mentioned to you before. So Phantom project is also one of the longest running functional genomics project. This is a project that is led by the Riken Institute in Japan, and mostly from the institute that's in Yokohama. And this project is actually mostly producing a specialized type of data, which is called CAGE, which means gap analysis of gene expression. So in the CAGE protocol, what you do is you target specifically rather than sequencing through RNA, you sequence the entire transcript, even you have to fragment it, right? With CAGE, you target specifically the 5' end of the gene, and then you sequence the 5' end of the gene with very short tags. And essentially what this is helping you is to identify the promoter regions of the genes and actually to measure the activity of the genes. The more cages, the more tags, the more active the gene is, and you know where the gene is being expressed, what is the promoter region. And essentially here they had a much more variety of cells, including blood that we did not have, and neural, and essentially we saw the same clustering, right? The endothelial cells, the epithelial cells, the mesenchymal cells, and then the neural cells that actually cluster together to the epithelial cells. And again, the same thing when we use not only RNA-seq based, but also RNA-seq based data, RNA-based data, CAGE and RNA-seq, but also when we use DNAs, so data, which means the regions of the genome that are open, the chromatin accessible regions. We see essentially this classification: endothelial, mesenchymal, epithelial, and then in addition the blood and the neural. So we actually concluded that there are, one of the things that we are concluding from this analysis is that there are 5 major cell types in the human body. So even though we may have hundreds or thousands of types and subtypes in the human body, what we claim is that they tend to belong to these 5 types, which are, again, epithelial, endothelial, blood, mesenchymal, and neuronal. And these more or less, right, correspond to the histological types that have been in textbooks for years, right? Like, but not exactly, right? Sorry? No, not exactly. So, is there a question, or sorry, or maybe not? No, okay. So that actually correspond to the classical histological types, right? The epithelial to the epithelial, but actually the endothelial, that is considered usually a subtype of the epithelial, for us is a proper type. The blood, that's sometimes considered a type of the subtype of the connective, is a separate type for us, and the sometimes connective and muscle are considered 2 different histological types. From the transcriptional standpoint, we believe that they belong to the same type. And we actually identify genes that are specific of each of these major cell types. These are genes that are expressed mostly, only or mostly in epithelial cells. These are genes that are expressed mostly or only in endothelial cells. These are genes that are expressed mostly or only on mesenchymal cells, right? So these are genes that, to some extent, define, right, these major cell types. And just for you to have an idea of one of these genes, this is a long non-coding RNA here. Here you see the annotation. This is a long non-coding RNA of a known function. This gene is next to protein coding gene here, right? And this is the RNA-seq tracks across all the samples that we had in our analysis. This protein coding gene here is highly expressed in essentially all cell types, as you can see. But the long non-coding RNA instead is highly expressed, but only right in cells of endothelial type, right? Cells of endothelial type. But as you can see, remarkably, it's not really expressed in a particular location in the human body. It's expressed everywhere in the body, in the aorta, in the skin, in the bladder, in the femur, in the lung, in the heart, etc., right? It's expressed almost everywhere, but only on these endothelial cells, right? And it would never appear as a tissue specific cell type, because it appears in many different tissues. What I just wanted to make is that we propose that these 5 cell types are like the building blocks, right? The building blocks that make, essentially, by combining in a different, and we will see this later, by combining them in different proportions with different subtypes, make the huge variability of organs and tissues that we see in the human body. Of course, as I said to you, one of the limitations of all these assays is that we are measuring the RNA based on the RNA extracted from millions and millions of cells, and therefore we don't have really a clear view of what is the transcriptome of an individual cell, but rather the average, right? The average expression of the genes across the population of cells that we are profiling, what we call bulk expression. As you probably have heard, that there is, in this area of functional genomics projects that are more and more have higher and higher resolution. There is the human cell atlas. The human cell atlas is an aim to produce a comprehensive reference map of all human cells. And as you can see, there are many cells in the human body. There are 10 to the 13. This is a lot of cells, right? It's a lot of cells. It's a billion using European nomenclature, right? It's more than 10 billion, 10 millions of cells in just one body. And the idea is to characterize all the cell types. So I think that the aim of the human cellology is to sequence about 10 to the 12, 1 billion cells at the end. That's really a huge amount of data, right? A huge amount of data. It's 1 billion RNA-seq data sets. Anyway, you are probably familiar. I mean, the things that these protocols allow is actually something that we could not do with the bulk RNA-seq that we were used to. This is to identify subpopulations. Because in the RNA-seq, if I was taking 50,000 cells from a breast, if I was going to do a bulk RNA-seq experiment, as usually done from these tumors, I would have just one number of expression per gene. I would have a single dot. Now I can sequence each cell independently, and then when I do this visualization using TSNEs and whatever things PCAs or many methods that are being developed to visualize these things, you actually see a high resolution by seeing these subpopulations of cells, and then many of these corresponds to known cells, but also you also discover new cell types that you did not know about. And I like to go back to this thing. So now that we did this encode thing was started before single cell technology has widely available, but of course now we had a lot of single cell data, and I wanted to see how robust is how robust is our prediction, right, of the 5 major types on using single cell data. Even though there is a lot of single cell data, there is no yet, right, multi-tissue single cell data from the same donor, which will be comparable to what we were doing with Tom Gingeras. There are many single cell data sets from many different organs, but done in different ways, using different protocols. Here we were able to build on data that has been existing on single cell, not in human because this doesn't exist yet in human, but on mouse, right? So in mouse, there is this data set called Tabula Muris, in which they had actually produced single cell of 20 different mouse organs. And when we cluster these cells, right, these are 53,000 cells that were sequenced using 10x. But actually, I think that you could still see that the cells more or less recapitulate our major types. This is blood, this is endothelial, this is mesenchymal. The epithelial always gets a little separated, and then of course we have this weird type. This is actually hepatocytes. Hepatocytes are typically considered epithelial cells, but they are really, if so, they are very, very specialized epithelial cells. And actually, when we do the clustering now this is the clustering of the cells. So the clustering of the 50,000 cells. This is the clustering of the cell types. The authors of this paper classify the cells into 75 cell types. When we actually do the clustering of the cell types based on the transcriptome, actually we recapitulate more or less, right, the major cell types. These are the blood cells, these are endothelial cells, these are mesenchymal cells, and these are epithelial cells, more or less, right? So we do think that the single cell data is sort of supporting this idea of a hierarchy of cell types, at the root of which are these 5 major cell types. So let me move a little bit into this idea. So remember that we are trying to follow up what happens after the sequence of the human genome 20 years ago, and what things, right, were triggered, right, because of our ignorance on what the sequence of the human genome meant. And as I said, we started first by trying to characterize human genome diversity, and then the second large thing was this idea of functional, of trying to characterize the function of the human genome. And I talk about the Encode project, right? About the Encode project. I talked already about the Phantom project, but these are not the only two important large-scale project. There is the roadmap epigenomics also in these are, of course, these two projects are from the United States. roadmap epigenomics, which is sort of similar to the ENCODE, but mostly focus on chromatin modifications and other modifications of the genome. And then in Europe, there is the Blueprint project, which is an ENCODE project, but focus on the hematopoietic system. So the idea was to characterize through functional genomics the different cell types that exist in the blood. And then there is this other project that I also like to stand a little bit more, which is here. Yes, the Ctex Project. Because the Ctex Project, to some extent, tries to bring together these two angles, right? Human variation, and human function. Because at the end, human variation impacts genome function. That's why people with different genomes are different, right? Changes in the genome, right, impact the function of the genome. Mutations in the genome, deletions, insertions, SNPs have an impact on the function of the genome, and the idea is to understand, right, how this variation impacts genome function. And this is essentially the first project in which I think this could be considered, that was being analyzed, is the Gtx project, right? The Gtx project is a project in which data was collected from about 1,000 post-mortem donors. So these were people that had died and from which the organs were extracted. Of course, they donated the body for this project. They did not, but their families did donate their bodies for this project. And organs were extracted up to almost 50, in some cases 50 organs per body, but on average is about 20, 25. And the RNA was extracted from these organs and it was sequenced, right? So we had sequencing of the organs of about 20, on average 25 organs of about 1,000 individuals, right? And actually the genome of these individuals was also obtained. So we have a database in which we have genetic variation, right? Because we have the genomes of 1,000 individuals, and we have some sort of functional activity because we have the RNA-seq, we have the gene expression on about 25 tissues on these 1,000 individuals, right? That's really a great resource. That's a great resource. So the main aim of this project is to understand essentially how genetic variation affects gene expression. So it's bringing together these two angles, right? Emphasize genetic variation and genome function. And essentially, I mean, the paper, well, I think the paper, well, I mean, I'll tell you, I think I have a picture of the main aim, as I said, is to identify single nucleotide variants in the genome that affect gene expression, what is called expression QTLs, right? Expression QTLs and SNPs in the human genome that, depending on the nucleotide, right? Depending on the nucleotide, on the genotype, you have a gene that is expressed in one level or another. Let's say individuals that have the genotype AA, the expression of the gene is very high. The individuals, the genotype of which is CC, the expression of the gene is very low. Individuals that have the genotype AC have an intermediate expression, right? That's an expression QTL. And the main aim of this project was to identify expression QTLs. This is a very, I'm not going to get into the details because this is a very complex project from the logistic standpoint. It involves the organizations that procure the bodies and perform the autopsies and the dissections. It includes the biobanks where the tissue samples are stored, and involves the center in which the sequencing of the RNA and of the genome is being performed. In this case was the Broad Institute in Massachusetts. And then, of course, involves a number of groups in analysis. And one thing that's also important, this project is not targeting any specific disease. So these individuals were not chosen because they had a disease. Actually, ideally, these individuals should be as healthy, of course, as healthy as possible when you die, right? Which is a sort of a contradiction. But anyway, But one thing is that, for instance, these individuals could not have cancer, could not have infectious diseases, have to be in some relatively good nomenclature, which means that you have a good aspect, you look good, more or less. And one important thing is that the postmortem interval, right? The time from death and organ extraction should be less than 24 hours to avoid any degradation of the biological segments that are occurring with death. And I will talk to this a little bit. And there was one important thing is that the images being taken from all organs. This is also important. And there is a pathologist that reviews this is to pathology images, right? This is actually things. So for instance, this is one of the samples that has been sequenced. This is an artery, and this is the pictures of the artery, the different sections of the artery, and you can see that the PRC comments means the pathology review comment, or PRC's pathology review committee comment says atherosclerosis with up to near total occlusion of vessel, Pacinian corpuscle identified, right? So there is something, there is information on these histological images. So as I said, this paper was actually very also the paper of the last phase of Gtx. Gtx also has 3 phases, a pilot, an intermediate, and a final. The final phase of Gtx has been published also a few months ago in Science. So this is the main issue. Here is where this paper about the genetic variation between sexes was published, and I will promise Rita that I will talk about this next year. Okay, this year I decided, because I think it's more appropriate, I decided to talk about death, since I think it's more consonant with the times. Anyway, this is the Ctex transcriptome to have an idea, right? So this is the number of individuals for which we have samples, right? So there are samples that are easy to extract from the individuals, these individuals that die, that are dead, and some of them die in accidents. So for instance, the blood is easy to extract, the skins are easy to extract, the muscle is easy to extract, and some other organs that are more difficult to extract, and some other organs that, even though they can be extracted, right, easily, like kidney, right, they are prioritized for organ transplantation. I mean, I don't want to enter into the details of this project, but this project is very complex from the ethical standpoint as well, right? So if you have someone that dies and has a kidney, I mean, you will not use this kidney to do RNA-seq. You will use this kidney to give it to someone that needs a kidney, which means actually that the organs that are most commonly transplanted, that kidney, I think lung as well, liver, sorry, not lung, liver here, right? These organs, right, are not very well represented in Gtx. And other organs that are not important in transplantation and are easy to reach, because you have to do this process of taking the organs from a body before 24 hours, and this process takes a few hours, and you have to start in a particular order. To reach some organs, you have to take out some organs first, etc., etc. Anyway, and on average, I don't see you see it very well, but on average, I think it's about 20, I said 25, but I think it's about 20 tissues per individual, okay? And just the data is very simple. Huh? I have to say, if you are studying bioinformatics, the data is very simple. The data is simply a table with the name of the gene and the expression of the gene in this sample, right? Another table with the name of the gene and the expression of the gene in the sample. So for instance, let's say this is the heart of one particular individual. We see, for instance, that the gene in the heart of this individual is very highly expressed, right? This number means that it's very highly expressed. However, the gene is very lowly expressed in the esophagus, maybe of this individual, or maybe it's a different individual. On the other hand, for instance, the gene is quite highly expressed in the intestine of this individual, but is not expressed essentially in the other individual. So we already see, or for instance, this gene is very highly expressed in the esophagus, and is actually also very highly expressed in the heart, but it's not very highly expressed in the intestine. So we see this variation of expression, right, between tissues and between individuals, right? That's what. But if we cluster the samples based on the values of gene expression, essentially we do the same thing that we did before, right? We just compute some sort of correlation between these two columns or some sort of distance or whatever, right? And then we compute how close two transcriptomes are, right? So that closer the dots here, more similar the transcriptomes of these two samples, right? We see that the samples cluster mostly by tissue, right? Samples cluster mostly by tissues. These are all the blood sample, and essentially the main division is, as was already known, is between non-solid tissues, right? We have here blood and LCLs. LCLs are cell lines derived from blood. And within the solid tissues, the main divisions are between brain and the rest of the tissues, right? And one thing I wanted to mention is that individuals don't look to play a strong role in this classification. I mean that maybe this dot and this dot and this dot correspond to the same individual, right? 3 tissues from the same individual, but these 3 dots do not cluster together. The contribution to the individual to the variation of gene expression is much smaller than the contribution of the tissue. So the same tissue from 2 individuals, in other words, the same tissue from 2 individuals is transcriptomically much more similar than 2 different tissues from the same individual. This is not surprising, obviously, because you can transplant a heart from an individual to another, and you can't transplant my brain to my heart, right? But still, it is reassuring that we see this from the transcriptional standpoint. But of course, that's the thing, right? This is a transcriptome of the death, right? This is a transcriptome of people that have been dead for hours. And the question is, how representative is this of the transcriptome of the living? Because we are not really interested in the transcriptome of the death in general, right? We are interested in the transcriptome of the living. So the way we approach this in this paper, which was part of the pilot phase of Gtx, was through the analysis of samples that have been taken from individuals that were alive. This mostly biopsies or sometimes amputations as well. And then we just plot everything together, right? In color, we have the Ctex samples, which are post-mortem. And in gray, we have the samples that are taken from people that are alive, ante-mortem. And as you can see, even though they don't match identically, you can see that there is a good correspondence, right? The blood samples ante-mortem cluster together with the blood samples post-mortem. The lung samples ante-mortem cluster with the lung samples post-mortem. The thyroid samples pre-mortem cluster with the thyroid samples post-mortem. The heart samples pre-mortem cluster more or less with the heart samples post-mortem, etc., etc., etc. So there is some sort of transcriptome signature that defines a tissue that is, to some extent, retained in post-mortem samples. But of course, this doesn't really mean that death doesn't have an impact into the transcriptome of the tissues. It has. Otherwise, that wouldn't have any meaning. If after the death of the individual, the transcriptome of all our bodies would work essentially the same, we will not notice that we were dead, right? Everything will be the same. And we actually were interested in this. We were interested in actually knowing, just to understand, right? I mean, our interest initially was to understand how well we could take the data from living individuals, right, from dead individuals as representative of the data from living individuals, right? So how representative? What should we do, right? How we should take into? Should we do something, knowing that the transcriptomes that we are investigating and transcriptoming for people that are dead when interpreting the results? And actually, the first thing that we saw is that there is a clear impact of the death in the transcriptome of the tissues, right? This is even though I would say that there is a signature that's preserved, this doesn't mean that there is no impact, okay? So this is actually the clustering of blood samples that actually, for some reason, blood samples some blood samples were taken pre-mortem, and some samples were taken post-mortem. I mean, let me tell you, this is not that people were waiting for people to die. They take the blood. So these people probably died in hospital conditions, right? And it may actually that because of the procedure, So the sample, there was sample taken before the individuals really were declared dead. And if we cluster the samples based on transcriptome profiles, we actually clearly see that there is a separation between the pre-mortem samples, right, and the post-mortem samples. So it's a clear separation. So actually, death has an impact on the transcriptome of the tissues. And we'll look at this into more detail, because the thing is that we have only 24 hours, but within these 24 hours, we have individuals with almost a continuous range of postmortem intervals. You know, we have 1,000 individuals. In some of them, the procedure started right away, and some organs were extracted less than an hour after death, up to 24 hours. So we have a range of postmortem intervals from individuals and for tissues, right? So this, to some extent, creates some sort of temporal trajectory, so that we have data at different times since death. And here, of course, what we have, these are, in the columns, are the individuals, and this is the postmortem interval of the individuals or of the sample, whatever. So these are individuals in which the liver was extracted 1 hour, not later than before an hour after death. This between 1 and 4 hours, between 4 and 6 hours, between 6 and 15 hours, and more than 15 hours, right? So and this is a number of genes. And you would expect the following, and maybe if you never thought about this, you say, well, the organism dies, therefore the genes start stopping work, right? And slowly the expression decays, because what is the point of having the genes being working in the cells when the organism is dead? But actually what you see is it's not exactly this, right? We do see red. I think that red means more expression, maybe the other, but doesn't really matter. So we see genes that indeed, right, they are still expressed within the first hours, and then after 6 hours they decrease expression. But there are other genes that actually are mostly silent, and actually as a reaction of the death of the organism, they get activated. So this is actually seen in this case, right? This gene here is an hemoglobin, and this gene, this corresponds to the same intervals than before. Sorry, it's less than an hour, 1 4 hours. Anyway, this is time. Let's say this is time since death, right? The more to the right, the longer the time since death. And you can see this gene, and these are different tissues. You see, the gene actually increases expression after death. And when you see the result, initially you are really confused, right? Because what is hemoglobin? Hemoglobin, the function of hemoglobin is to carry oxygen. So what is the first consequence of the death of the organism? The first consequence of the death of the organism is that oxygen doesn't arrive to the cells anymore. The lungs stop, the heart stops beating, the lungs stop breathing, and the cells don't receive oxygen. So why then, if the cells don't receive oxygen, you actually get a systematic increase, right, of hemoglobin in many, many, many tissues? Well, the hypothesis is maybe because that's essentially what it should be. So there is a sense, the cells sense the lack of oxygen. The only way in which they can react, of course, they don't know that the organism has died, right? So they only sense the lack of oxygen. The only way in which they can compensate the lack of oxygen is actually by producing more of the molecules that carry oxygen, as a way of this, what is the name of the word? Lack of oxygen. Anyway, to hypoxia. It's as a reaction to hypoxia, right? This is a reaction to hypoxia. The only way in which the cell can react to something that is damaging the cell is to generate more of the genes that bring oxygen to the cell, right? Because the cell instinct, the self-determination is to live. Similarly, in this case, right? This is also an interesting case, which is also initially counterintuitive. This is RNase. This is an enzyme that's involved in the degradation of RNase. So you would expect, well, the organism is dead. Cells are not dead anymore. RNA is not needed at all. Why you should actually, if any, you should increase the RNases so to degrade the RNAs. But actually, it's the other way around, right? Again, there is a sense that transcription is not proceeding, that there is a damage being produced to the RNA, and essentially the only way the cell can react is by shutting down, right, the mechanisms that the cell possesses to degrade the RNA. And as you can see, RNase is again one of the genes that decreases expression more constantly across multiple tissues, right? So essentially, what this is telling us is that the death of the organism triggers a number of the death of the organism doesn't really mean immediately the death of the cells. The cells react to the death of the organism, and they do so in a quite tissue-specific manner. Here, again, this is time after death. Here we have tissues, and here in colors we represent the number of genes that change expression with respect to the previous time at these time points, right? And as you can see, there are tissues that react immediately, that change the expression of many genes right after death. And the most notable case is muscle, right? In muscle, there is a huge changes in gene expression, a huge impact in the transcriptome right after death. Then there are a number of tissues that have a sustained response, right? They have a sustained response. They change expression more or less continuously during all this 24 hours interval. Of course, we can go beyond 24 hours interval. And then there are other set of genes, right, that actually had a peak response, mostly at 6 hours, will actually change expression. They remain more or less stable. They are able to support the death of the organism for 6 hours, but then they start changing, they start reacting. And then there are genes that react very late, but they are very stable. They react very late to the death of the organism. So one thing that we did more as an academic exercise, what we did is to say, well, since we know that genes change expression after death in a way which is specific of the tissue, could we use this information to predict the time since death? You know that this is still a problem in forensic medicine, right? You've seen in the movies when the police gets to a crime scene or to a dead body in accident, they always try to determine how long has passed since the individual passed away, right? And they use different things like body temperature, rigor mortis, etc. It's still not very precise. So we were speculating, could we actually go to the body, do some extraction of RNA, do RNA-seq, and from the values of expression of the genes in this tissue, could we estimate, could we infer for how long this individual has been dead? And we use some machine learning methods, gradient boosted trees. This is the work, actually, I say Federation de Patho, but actually this is mostly the work of Manuel Muñoz, a very talented student in my group. Sorry, because I don't like this. I think that this is mostly the work of Manuel Muñoz, so I want to put it here. We actually got some relatively good predictions. So we were able to, just based on the analysis of the RNA-seq in multiple tissues, although we actually tried to minimize the number of tissues that were needed to get good predictions, we actually could get relatively good predictions of the time since death, right? They were better. These are actually minutes since, these are the real minutes since death, right? This is in minutes. This is the predicted postmortem interval in minutes. And when there were not so many minutes, like let's say, I don't know, 8 hours or so, or 10 hours, our predictions were not bad, but the predictions became worse as longer the time since death, which in any case, we never claimed this to be any effective method for predicting the time of death. For some reasons that you never know which of your papers is going to have a higher relevance. This this work actually took catch the attention of many media, and it was in many places all over the world, right? So there was actually a lot of interest in this thing that was really very minor in our paper, because our main interest, as I said, is trying to understand how reliable were the measures taken on deaf people as representative of the measures that could, if possible, be taken in deaf people. And actually, there was even some sort of philosophical comment in our work, right? If your cells continue to function, what does it mean to die? And all these things. But actually, I think there are some interesting reflections in multiple directions about that, what death means, and biological death means, and what is actually being an organism, and what makes us. I know that some people, maybe within this room, believe in the soul of all these things, right? So it's an interesting discussion. Anyway, as I said, yeah, let me continue with Gtx. We actually use Gtx for another. I'm getting maybe a little bit too long, but I will finish soon. Anyway, so remember that we had these things in Gtx, right? That we know that the tissues, right, show a characteristic signature, and you know that these tissues, each of these tissues, as I show, because our encode work is made of cells of very different type. Right? You see the lung, the primary cells from the lung were everywhere. And remember that we actually identified genes that were specific of these cells. So the problem that we of these major cell types, right? Remember that we have the major cell types. So the problem that we were trying to address was the following. Even though we just have from one Gtx sample, right? We only have one experiment. Therefore, we have a value of expression per gene, and we don't know to which cells correspond. Can we infer what is the proportion of cells of different type from this? So from this bulk tissue transcriptome, can we infer the cellular composition? And actually, this is a very active field on transcriptomics and bioinformatics of transcriptomics, because actually you can, well, there are many methods that try to predict what is the cellular composition of an organ based on the bulk expression estimated in this organ or in a tissue, or whatever, on a heterogeneous population of cells. And the intuitive idea is very clear, right? So this, as I said, these are genes that are specific of these cell types, right? Let's forget about, let's say, epithelial, endothelial, and mesenchymal, right? Now I have my expression, right? Let's say I have expression, let's say, of skin, right? That is skin. I have expression of the bone, right? And I have these two things. Now, how can I infer what is the proportion of epithelial, endothelial, and mesenchymal cells in those two bulk transcriptomic data sets? Well, the idea is the following, right? If I found the genes that are from epithelial, that are specific of epithelial, that are found only on epithelial, very highly expressed in the skin and very lowly expressed in the bone, I would infer that there are more epithelial cells in the skin than in the bone, right? If I actually found that the mesenchymal cells, the mesenchymal genes, are very highly expressed in the bone, right, and are lowly expressed in the skin, and then I would actually assume that the bone have more mesenchymal cells than the skin, right? So the idea is that based on the patterns of expressions of genes that are specific to a particular cell type, you can try to estimate the proportion of the cell type in a sample in which you have done bulk transcriptomic analysis, bulk RNA-seq. And there are many methods. Anyway, I just wanted to say that this is what we did with the Gtx tissues, right? So we use these sets of genes that are specific to our 5 major types, and then we attempted to estimate the composition of the Gtx tissues, right? So these are the different Gtx tissues, right? And these are the different, our 5 major different types: endothelial, epithelial, mesenchymal, neural, and blood. And then, of course, this is the distribution of our estimates across the 1,000 individuals, or whatever individuals of which we have. And I think things makes a lot of sense, right? For instance, these are enrichments. This is a method called Excel. Sure, I didn't put it here, I'm sorry. So essentially, the brain regions from Gtx are enriched in our neural cell types. The blood from Gtx is enriched, the blood cell type. And if you look actually at the enrichments in the different tissues, they tend to make sense. Let's see this thing. This is, for instance, the esophagus has two layers. One is the mucosa layer, right, which is the external layer, is an epithelial layer, and a muscularis layer, right, which is mostly muscle, that in our classification is mesenchymal. And as you can see, as expected, the mucosa region of the esophagus is enriched for epithelial cells, while the muscularis region from the esophagus is enriched in mesenchymal cells, as you would expect. So we think that we get some sort of good estimation of the composition of the human organs based on these four major cell types. And actually, when you put them into, I think that they should turn, but it's not turning. Anyway, I mean, when you just cluster the Gtx tissues based on these proportions, we really get a very good representation, a good separation. I mean, a good separation of the tissues. So essentially, what we conclude from here is that the broad cellular composition defines tissue type, and maybe that alterations in this composition might reflect tissue histology, morphology, and pathological states. And the way we actually tested this hypothesis is by actually resorting to the Gtx images. As I said, the Gtx images, it's a set for each sample profile in Gtx, there is an histological image, right? And the histological image it's annotated by the pathologist, right? By the pathologist. So the idea is the following: can we see in the histological images a reflection of the changes in cellular composition? And that's very difficult, right? Because dealing with histological images is complicated. We're working on this, but I will not show results about this. But maybe it's easier to work with the pathology and with what pathology sees in these images, right? Anyway, it's not actually not easy to. It is not easy to work with this either, because this is written in free format, right? Let's say that we want to. This is testis, right? This image corresponds to testis. Let's just say that I want to separate testis in which there is strong spermatogenesis from those in which there is absent spermatogenesis. I cannot search for the word spermatogenesis because some pathologists may have written no spermatogenesis or reduced spermatogenesis or mild spermatogenesis. Others might exist strong spermatogenesis, active spermatogenesis, etc., etc. So there is the need, right? The need for processing these natural language sentences to make some sort of control vocabulary of what we call the histological phenotypes in Gtx. And this is what Manuel Muñoz actually did using some linguistic methods. And at the end, we end up with a number of what we call Gtx histological phenotypes that actually classify the Gtx images, right? Some of these phenotypes, like for instance congestion, are common to many tissues, right? This could be lung congestion, there could be liver congestion, kidney congestion, etc., etc. But some others, for instance, like spermatogenesis, affect only one particular tissue, essentially only the testes. And then we look whether we did compare the following. We compare the samples, right, that were annotated as having one of these phenotypes with the samples that did not have this phenotype. So we compared samples annotated as atherosclerotic and samples without atherosclerosis, let's say in artery, right? And we compare the cellular composition of these two samples, right? So let's say we have, again, I repeat, we have artery annotated as atherosclerotic and an artery not annotated as atherosclerotic. I grouped all the arteries that are notated as atherosclerotic and all the arteries that are notated as no atherosclerotic. And then I compute the estimated proportion of my primary cell types in each two groups and compare and see whether there is a difference in the composition. And actually, this is what I see, right? When I compare my proportion of estimated proportion of primary cells in arteries with atherosclerosis, I see an elevated proportion of endothelial cells compared to normal arteries. Or when I look at the skeletal muscle, I see an elevated proportion of mesenchymal cells in the atrophic muscle compared to the normal muscle. This is atrophic, normal. Or actually, when I look at the breasts in males and females, I see actually strong differences between the composition of male and female breast. With females have enrichment of epithelial cells, which is consistent with the ducts that produce milk, compared to males. Interestingly, there is a condition called male gynecomastia that actually it occurs in older males that tend to grow breasts similar to those of females. And as you can see, males with gynecomastia had a cellular composition that's very similar to that of females, which mirrors the histological findings. And actually, we also saw changes in cellular composition occurring with years. So this is your lungs, most of you. This is my lung of an older people. And as you can see, what happens with age is that we get a reduction of the epithelial composition of the cells in the lung that occurs with age. One area in which we expect larger changes in cellular composition is in cancer. So we went to the international consortium, international ICGC, International Genome Consortium, whatever, ICGC, International Cancer Genome Consortium, ICGC, where they have produced RNA-seq data from many, many cancers. And here what we are comparing is the cancers, right, with the normal samples. This is from Gtx. This is the normal controls. I'm very happy that the behavior of the normal from the cancer projects and the normal Gtx is very similar. Here we are plotting the proportion of endothelial cell types, and as you can see in many cancers, in many kidney cancers, right, there is an increase in endothelial cells, which is consistent with the vascularity observed in most types of cancers, but actually in this one, which is not observed, and this is known. I forgot the name of this cancer. And actually, when you compare within each donor, right, the cancer, the tumor, and the normal sample, you in general see an increase in the endothelial cells in the kidney cancer. This is not only comparing cancers versus non-cancer. This is ovarian cancer. In this case, we see this is normal individuals without cancer. These are individuals with cancer. There is a huge increase in epithelial cells in ovarian cancer, but actually the increase is even larger when you move from primary to recurrent. So there is a change in the composition of the tissues, which also indicates the stage of the cancer. Let me just. Oh, I'm going to the end of my presentation. I think it's very long. The end of my presentation. So I think that it's very. I mean, what I'm trying to illustrate with these examples is that here we are relating molecular data and phenotypic data in histological images, and I think this is very important, right? This is another area in which there will be the need for great developments, right? Is that when you put together multiple both molecular and phenotypic data, clinical data is when you start learning. And in this case, I think that one of the best resources now in the world is the UK Biobank, which they are following 500,000 volunteers, and they are actually had a lot of data that they have collected on these individuals, and actually they are now complementing with genotyping and some actually functional genomics data as well. On this line, right? So on this line of enhancing the bringing together the variation angle and the functional genomics. I like to talk to you just one slide about the Ctex Project. The Ctex Project is a consortium between the Gtx and the Encoding Project. And essentially, remember, in Gtx, we had multiple individuals, right? And one assay, RNA. In Encode, we have multiple assays, but we have ChIP-seq, RNA-seq, Hi-C, DNAs, ATAC-seq. We have multiple assays, but we don't have individuals. Now, the Ctex, not tissues. The Ctex project tries to bring everything together, right? That's really a very interesting problem in terms of personalized medicine, right? Personalized medicine, because this project is taking 4 individuals, right? Multiple tissues from these individuals and doing multiple assays, right? So it will be the first time that we will have this data available. We will have multiple assays from DNA, RNA, chromatin interactions, opening of chromatin histone modifications, DNA manipulation, etc., right? In multiple tissues, in multiple individuals, right? So this is probably the way to go into the future to try to understand how genome variation impacts genome function, right? Impact genome function. Okay, let me finish now with the last, very briefly this, because this I don't have any data here to show, just to let you know what's going on in this area, in the other area. So I'm talking about, as I said, remember, we have 20 years ago, just this week, the Human Genome Project, how this has evolved, right, into multiple areas of post-genome research, human genome variation, functional genomics, now the sequencing of organisms. After the human genome sequence, after the sequence of the human genome was obtained, the sequence of other organisms was also sequenced, and the priority was given actually to the model organisms. In fact, the model organisms, as you know, are fly, worm, mouse, rat, zebrafish. I think these are considered the main model organisms. Anyway, so actually, even prior to the human genome sequence, the fly genome was published before, but then after the human genome sequence was obtained, there was the mouse, then the rat, the zebrafish, and then many organisms that are of interest to humans in terms of food, like the cow, like the chicken, and then the plants that feed the rice, the tomatoes, like the beans. So all these were given priority. I'm just bringing data from this. I'm bringing data from this slide, which is a little bit, the paper, which is a little bit old, because this paper is from 2015, right? Let me skip this one. So in 2015, there were about 5,000 animal and plant species sequenced. So I don't know actually how many species have their genome sequence, but I can tell you, you can check if you want. I'm going to make a bet. So this doubles every, let's say, 1.5 years, right? So in 2015, there were 5,000 species. In a 1.5 years later, at the bit of, let's say, 17, there were 10,000. And a 1.5 years later, let's say 19 or something like that, 20,000. So now we may actually have 40,000 animal and plant genome sequence. Okay. Now there is a project, which you may have heard or not, which is called the Earth Biogenome Project. This is an international project, which is a confederated network of networks that actually aims to sequence 1.5 million non-eukaryotic species in 10 years. This is not so far off, because this project started like 2 years ago. And if you just do this idea of doubling every 1.5 years or so, it will actually, even though now this seems impossible, right? Because, I mean, how do you locate the 1.5 million? What the logistics of this project is even actually more complex than any other, because I mean, the human cell atlas and all these things, you have many, many millions, right, of data points, but more or less everything is controlled. But here, the species live everywhere. Some species are located only in some remote places, so that's a huge undertaking. Anyway, this is a project, as I said, that's now being taken place. Actually, Montserrat Coromines, who was going to speak tomorrow, I think, or Thursday, but she cannot because she is sick. But I think she will be able to present in the next week or the other. She's leading. I'll tell you later. So these are nodes that correspond to taxa. So there is the vertebrate genome project. There is the 1,000 birds. There are the arthropoda, the beetles, but there are also national nodes, right, or regional nodes or geographical nodes. So the most advanced of all is the Darwin tree of life, which actually aims to sequence the genomes of about 60,000. Well, now it changes, say 60, now 70, whatever 70,000 species that lives in the British Isles, right? In the British Isles, and this is the more advanced project. They already have a few hundred genomes already sequenced, and they are actually putting together on all the infrastructure to make this possible. Actually, as I said, Montserrat Colominas is also the president of the Catalan Society for Biology, and actually this society is spreading a project which aims to sequence all the genomes of the species that live in the Catalan territories, which is about 40,000, and we have already about 25-30 genomes being sequenced under this project. Catalonia, it's a diverse hotspot. About 25% of the European species are found in Catalonia. But there are a lot of activity. Actually, there is now something called European Reference Genome Atlas, ERGA, which is overarching aim is to sequence the genomes of about the 200,000 species known to live in the continent. So I think I will finish here with this. And again, I mean, I focus mostly in one area, which is the functional genomics, but I wanted to think that there is a lot of research in multiple areas of the post-genomic times that will generate many, many challenging problems. For instance, and this is something that our friend, I don't know, maybe did Cedric already teach to you or not yet? Cedric Notre Dame.
On Friday.
He will, I don't know, maybe he will explain about this, right? But of course, let's say that we sequence 1.5 million species. There are gene families that have 500 in the represented in the genome, right? I know some kinases or something like that. Let's say that you want to build the tree of all kinases on Earth. This will represent 500 million sequences. There are no methods to align 500 million sequences. So Cedric Notre Dame is actually one of the few working on methods to produce this huge, large. So there is, I mean, although I said I did not focus on this of the sequencing of many species will also opens huge amount of opportunities for all areas of bioinformatics, from structural biology to sequence alignments, to functional genomics, to phylogenetics, to anything, right? So opportunities are multiple. So you will need like 40 Bologna summer and winter schools to train the people needed to deal with all these problems. And I like to stop here. So, I mean, this was one of the last times that our group could get together. I think this was already 2 winters ago, because we were planning to go last winter, but we had to cancel our group trip. This is people that participated in some of the work that I did. The Gtx work is mostly in collaboration with a group of Christine Ardile and Marta Melé and Pedro Ferran were the ones doing this work, as well as Manuel Muñoz, who is not here. And the work with Tom Gingeras was done by Alessandra, also Manuel and Valentin Pujers in particular. And I just wanted to thank, of course, all this work has been financed by the National Institutes of Health. And I wanted to also thank, I always like to thank this in my talks on the Gtx, because these are the people, the families of the people that donate the bodies of the donors, right? These are the people that agree, that consented to the bodies of the people, that their families that have just died, to consent for this project. And this is, I have to understand that this is very difficult, right? When someone dies, I mean, you can rationalize that maybe you're going to give the body for organ transplantation, but to rationalize that you're going to give the body for RNA-seq when probably 0.0000001% of the population knows what RNA-seq means is very difficult. And so the thing is, because these people actually, This is very American, but we can learn a lot from the Americans. These people actually came to some of the Gtx meetings, right? Because they actually wanted to see what was going on with the bodies of the people that they once loved, right? And I think that was very interesting, and I like to thank these people. Anyway, so I'm sorry because I think I took very long, but I'm still happy. Maybe I'll just stop sharing this, right? So that we can see my share. I'm still here, yes, and I think that some of you are still here. So if you have questions, I know it's late, it's time for lunch, but I'm happy to respond now or by email or for whatever.
Thank you very much for the beautiful lecture. Very comprehensive, I think, very exciting. There is a question from Rita, and I invite other people to raise their hand.
No, please. I would like the students go ahead of me, but I was raising the hand because finally I found the bottom after 4 times.
Okay,
so I have a question myself, huh? Okay, good.
Tamara Pertegella can ask the question.
Good morning. So thank you, Professor Guigò. I have a curiosity about the possibility to identify the proportion of primary cell types into the different tissue. And I am wondering which may be the impact of that in medicine or in research in general.
Well, I think as I show in some cases, for instance, we have conditions like, for instance, I showed the case of atherosclerosis, right? Or muscle atrophy, that, I mean, there is an alteration, right? When you have a condition that, in this case, affect the tissue, let's say, for instance, I know you have cirrhosis in the liver, right? So this certainly will affect the cellular composition. So you could actually think in two ways, right? If you see alterations of the cellular composition, they could indicate, right, that there is an underlying pathological state. On the other hand, actually, it also helps you to understand what are the cellular mechanisms underlying the disease, right? You see that maybe when there is cirrhosis, maybe there is an increase in maybe mesenchymal cells. I'm just making this up, but it's possible because, well, most cells in the we didn't look into this because most cells in the liver are hepatocytes. I don't know what they compare to when there is cirrhosis. I think it would be interesting. But I think it's both, right? It could actually be maybe, right, could be used to diagnose the status, but it's mostly to understand, right? I think it's mostly to understand how cellular changes impact the histology of the tissues. I don't know if I answer your question or not, but that's what I understand.
So just another curiosity. Do you think that being able to identify those changes in cell type composition would be more informative than doing RNA-seq or study about the mutation themselves?
I think it's a complementary thing, right? So you're right. I mean, if you do RNA-seq, you will see already changes in gene expression. Therefore, these changes in gene expression may be enough to indicate some changes. But sometimes, right, the changes of gene expression are mild or not significant enough, right? So you get, for instance, I don't know, 10, 15 genes that are, and you look into them that are differentially expressed, right? And they individually, they don't tell you anything. So that's what actually when you have a differential gene expression analysis, you run some sort of pathway analysis, right? That you see whether the genes that are mildly differentially expressed, they tend to belong to some sort of common biological pathway or process, right? So that you say, well, even though individually they are not very different, very strongly differentially expressed, taken together, right? They may actually indicate some sort of underlying condition. So that's the same thing. So let's say that we just have very few genes that are differentially expressed, or there is an increase on the expression of a few genes, but all these are of endothelial type. This will indicate, right? We will not see this maybe by doing differential expression analysis, but by actually estimating the proportions of the cells, we may actually see, because we are building into the information that are providing hundreds of genes, right? We may actually be able to infer that there is a change that goes into a particular direction that may be more informative to us, a particular It's not now that a particular gene is the responsible of the disease, but it's a particular types of cells that are involved in the disease. And this may, in some cases, be useful.
Thank you. There is a question from Federica Veneziani.
Yes, thank you so much for your talk. I have a question. Because in Gtx data, we have a large range of age of the people in the sample. What is the impact of the age on the transcriptome? And if we need to analyze the data using Gtx or with, for example, WGCNA, would be appropriate to analyze independently the different age groups? Especially if the topic, for example, is the metabolism?
Mm-hmm. Well, there is a wide range of age, yes and no. So for a number of reasons, the age in Gtx for participants is between 21 and 70, right? That's the age. So most transcriptional changes in the human body are likely to occur before 21. They are more likely to occur during the early years of development, right? That's when most changes are going to occur. Probably at 21, your transcriptome is quite well established. In addition to that, and I think it's good that it is this way, but of course it complicates the analysis. There is not a uniform distribution of ages of death, right? There are less young people dying than older people dying, right? There are people of 20, 21, 23, 24 that are in Gtx, but they are underrepresented. There is a bias towards the end to the older ages in the distribution, which is what you as a human being like, as a scientist don't, right? But we are human beings before scientists. So that's the thing. So there is a bias. Still, there are many analyses that you can do trying to understand how gene expression changes with age. We actually did this analysis, and we found, for instance, that among the genes that change most expression between age, there was an enrichment of genes involved in Parkinson and Alzheimer pathways, right? So these genes seems to change the expression with age. Now, usually, when you do analysis with Gtx, say you do, for instance, you want to do female versus male, or you want to do the QTL analysis, or you want to control, let's say, for instance, right, atherosclerosis versus no atherosclerosis, which is heavily confounded by age, right? Older people have atherosclerosis. So you usually, in your statistical models, you use this as a confounding factor, right? You put age, essentially, in almost any analysis that you do in Gtx, you put the postmortem interval, the sex, the age, the body mass index as confounding factors, because these are confounding all your analysis, right? So, I mean, you're right that you have to, when you're doing proper analysis, and unless you are specifically interested in age, you have to control for age, because otherwise, because they are much older people than younger people, right, in Gtx, right? Yeah. And because older people tend to have atherosclerosis and younger people do not, this is totally confounded. Totally confounded.
Thank you so much.
Thank you. Next question from Gabriele Vazzana.
Yes, can you hear me?
Yes, I can hear you, and I can see you, which I like.
Yes. So I'll start curiosity just to know, in the slide where you showed us changing in genome expression postmortem, I was looking at the fact that the brain cortex cells were in the slow changing region, so cells that the transcript changes occur more slowly. And I was just curious about can we say something about that? Some interesting information, some curiosity.
Yeah, unfortunately not, for one reason, right? It's just a pure technological reason. Actually, I shouldn't even show this thing. The problem is that in Gtx, the heads and the brains were treated very differently, right? After I know that's what it grows, but that's the way it is, right? The the biobanks were mostly on the northeast of the United States where the project took place. Most of the donors are from the northeast, but actually during the autopsies, the heads were cut and sent to Florida, to the Miami biobank. So they had to be preserved in a different medium. I think they were in liquid nitrogen. So the postmortem intervals for brain regions, I don't think are very reliable. So I don't want to say anything about the brain, and I shouldn't really take this because we are unable to really control actually because I don't really know what the postmortem interval means in the case, because they have actually very large postmortem intervals and the brain region. I'm not sure we include this into the paper. I think this is an old figure. I think that this was not included because I don't think that we should include the brain because of this reason. I'm sorry. I think it's very interesting, right? I think that's really a key question.
Now I feel better because I had approximately the same question.
Yeah, no, I know.
The idea is, if you have electrophysiological activity in your brain, even 15 hours after mortem, after you died officially, this would be contradictory because one of the possible parameters that I hope they include to declare that you are dead, it's indeed the electrophysiological activity of the brain. So now let's make this in this very simple way. Gene expression and electrophysiological activity of the brain, how do they correlate? Because if I have gene expression, then I have also electrophysiological activity, and maybe I can see myself dead. Well, it's a little bit fun, with a little bit of fantasy.
Before time,
so,
right?
if you are saying what you said, and I appreciate it, tonight I can sleep. Otherwise, I wouldn't. Okay, good.
No. The thing is that I don't want to say anything about the brain. I think it deserves a specific study that we didn't do. Because we need to look a little bit more in detail what is the postmortem interval values mean for the brain regions, because they were treated very differently and they have very long postmortem intervals, and we don't understand actually this. So I don't know whether the brain is very stable transcriptionally or degrades immediately. I cannot answer this question. I could say that in our hands, the muscle changes very rapidly because we see this, but the brain, I cannot say that is very stable as it looks like in the slide that I sent, which I think it's a little bit misleading.
And if I may, although I couldn't do this question, and the idea is, are you going towards astrophysics, or it's an impression that you have a telescope behind you?
No, actually, I'm very bad. I like a telescope, but the only thing I can watch with the telescope is the moon. I am a very bad astrophysicist. But it's actually beautiful. If you haven't looked at the moon by a telescope, it's really amazing. See?
Okay. Sorry for the question.
It's just with my son. It was my son, but when he left the house, he left the house, but had us to keep the telescope.
Because I think that our hope is the sky. In any case. In any case, thank you so much for this wonderful lecture. Other questions?
More questions from the audience? Luca Leomazzi asks if it is possible to have a list of articles cited during the lessons. But okay.
Yes, I will send them to you, yes.
Thank you very much. More questions? If not, I thank Roderic very much for the beautiful lecture. I remind everybody that tomorrow morning the lecture of Professor Corominas is cancelled. We hope to reschedule later. And so the next lecture will be given tomorrow in the afternoon, starting from 3 in the afternoon, by Professor Graziano Pesole, who would talk about comparative and evolutionary genomics of SARS-CoV-2 data resources and open science. So thank you very much to everybody.
Thanks.
So, thanks, Roderic. Bye-bye.
Thank you.
Thank you.
Next year.
Thank you. Bye-bye.
Bye-bye.
Hoping to see you soon. Bye-bye.
Bye.